Perez. I work at the Paul Fraser's lab in Royal Holloway in London, and I specialize in metabolomics analysis, especially in plants and microorganisms. And so the work that we are presenting today is linked to this work, and is related to the Wi-Fly and RTV Foods project. And it's about capturing the metabolomic diversity in the cassava crop. So just briefly introduce the technology. Metabolomics is defined as the comprehensive study of all the small molecules in a living organism. Is the closest link to the phenotype within the context of the system biology. And the study of the metabolome is relevant for understanding the connection between the genotype and the phenotype that is observed. So over the years, we have developed a number of analytical workflows and different methodologies for understanding and studying the changes in the metabolome. So these methodologies are organized in two different strategies. The main one is based on an untargeted profiling of all the chemical features that are present in the plant extract are taken into consideration for the statistical analysis. And then only those ones that differentiate the different genotypes or the different treatments under study are moving to the identification pipeline. And that enables us to build customized libraries for specific crops, and also to map those changes into biosynthetic pathways, and also to integrate the information with other omics data sets, so we can establish correlations between metabolites and genes or transcripts. And the second approach is more specific for specific applications, and it covers targeted analysis of specific sections of the metabolome. So one of the main applications in where we have applied the technology for, especially for the RTV project, is to assess the chemical diversity of the germplasm collections. So this was initiated by my colleague, Margit Trappal, and she analyzed the in vitro material of a subset of South American and African varieties, and she established links or correlations between the chemical composition and the agronomic traits. And so later the project was escalated to a larger collection, and the objective in this case was to assess the stability of the metabolome over the years by comparing the material collected between 2016 and 2020. We concluded that a very small number of chemical features change over the years. Only 5% of them significantly change, and those changes affect a very small proportion of the genotypes of this collection. Also, the diversity of or the biological diversity was much higher than the variation introduced by the environmental effect. The second objective was to assess whether the chemical fingerprinting could be used as a classification tool. And by doing the hierarchical clustering, we can establish groups of genotypes that have a similar chemical composition. We are now in the process of correlating these with the phenotypic traits that they present. But doing the scatter plot, we can quickly identify what are the biochemical processes responsible for the phenotypic traits of these groups or these clusters of genotypes. And so moving now into the Wi-Fly project, and in line with the diversity of germplasm collection, we also analyzed a panel of East African elite varieties that were built during the 5CP project by NRI. And the interest in this collection of potentially in Wi-Fly and virus resistance. And so we analyzed this panel and observed in the PCA that the lines cluster according to the geographical location or the adaptation to different agroenvironments, and instead of the phenotype that was reported. We also noticed a lower diversity from the chemical point of view. A very small number of chemical features could differentiate the different clusters. We also noticed a high level of duplication in the collection. And that was also correlated or validated with the genotyping analysis. Within this objective of the Wi-Fi project, we also analyzed another panel of potential parental lines for breeding Wi-Fi resistance. This panel was selected based on the phenotype they presented under control infestation trials with South American Wi-Fi and with the African Wi-Fi. And the analysis of this panel indicated that ECU72, which is the most resistant one and one of the susceptible ones, called 2246, presented a distinctive chemotype compared to the other lines. And they were therefore selected for moving into the breeding program, but also for elucidating the mechanism of resistance in these lines. So by comparing the leaf material collected at different time points over the infestation with Wi-Fi, we could see the changes in the metabolome were constitutive, and the most significant changes were linked to the biosynthesis of monolignons, which are the building units of the lignin. That was especially evident in col 2246, in the susceptible one, that accumulated high levels of the intermediaries of these monolignons. The changes in the metabolome also were complemented or also correlate with the differential expression of the biosynthetic genes of this pathway. And this was done by Linda Welling team in UCR. So the other objective of the Wi-Fi project was to create a pre-breeding material and to create populations by crossing ECU72 and col 2246. So regions, QTL regions could be identified, and markers, biomarkers could be identified for elucidating the Wi-Fi resistance. So this first population produce a number of siblings with enhanced Wi-Fi resistance. And the chemical profile of this population revealed two different clusters within the resistant siblings. One is still very similar to the parent, but another one completely different than the other siblings, the other resistant siblings. So that could be an indication of other mechanisms present or other mechanisms segregating or epistatic effects, and that was corroborated or was in line with the conclusions of the QTL analysis conducted by SIAC team. So the following step was to make the decision of which one of these lines could be selected for advancing into F2s, and the discriminant analysis of this cluster indicated that CM199 and CM246 were able to explain the variation between the phenotype classes, and they were therefore selected for for creating F2s. But the discriminant analysis also enabled the identification of a number of chemical markers that differentiate both phenotypic classes, and we found 225 chemical features that highly accumulate in the resistant siblings. And we are now in the process of confirming the identification of these chemical markers, but the putative identification so far reveals a wide variety of chemical classes involved. We can see flavonoids and phenylpropanoids linked with the monolignol biosynthesis as previously found, but we also found a number of terpenoid compounds probably belonging to the sesquiterpenoid or apocarotenoid families, and a number of cyanogenic glycosides also involved in the resistance. So in summary, we have seen how the analysis of the small molecules can correlates well with the phenotypic traits, and how the technology can be used for complementing the assessment of the genetic variation in the gene banks collection. Within the Wildfly project, the analysis of the metabolome facilitates or complements the decision making process of selecting the adequate material for breeding. It also provides information about potential biomarkers that correlate with the phenotype and the genetic regions that confer resistance, but it also contributes to the elucidation of the mechanism at biochemical and genetic level. So I just want to briefly mention that there are other sources of pest resistance that we are working on. So the Peruvian lines, there's a number of Peruvian lines that also present high levels of resistance and a completely different mechanism than ECU72, and that opens the possibility of stacking or combining different traits for improving the resistance to whitefly in cassava. So the technology is in place and we have achieved some of the goals so far, but we think this is still a lot of improvement in some of the resources, especially the databases, the genetic and the chemical databases. We definitely need a bit an improved annotation and an improved curation of these databases, and that will help have an impact in the when it comes to integrate the different omics data set, and that will facilitate the exploitation of the untapped diversity that is holding this in the gene banks. Another dimension that the metabolomics analysis provide is an understanding of the whole changes in the changes that are occurring in the whole metabolome for accommodating the biosynthetic sections of the metabolome that explain or that makes the phenotype distinctive. So I would like to finish by thanking all the people that has been actively involved, especially my colleagues in Royal Holloway, Margit and Elliot, that have contributed to the establishment of the technology in the group, and our leaders, Paul Fraser and Jenny Fisi. None of this would have been possible without the collaboration with UCR and UCR teams led by Linda Walling and Luis Becerra, contributing with the material, but also sharing the knowledge about cassava biology and cassava genetics especially. All the teams involved in the Wildfly project and the coordinators of the project, John Colvin and Maruti, that have also participated in the design of the experiments and the data discussion. And of course, our donors, the Gates Foundation and the CGR centers. So thank you for listening. And